In this video, we're going to learn about translation or protein synthesis. So far we've learned about replication. So that happens in the nucleus and we make a full copy of the DNA. Then we have transcription, which is where we copy one gene into messenger RNA form. And that also happens in the nucleus. And then the final stage in protein synthesis is translation. So we're converting the mRNA now into a protein. We'll do a little bit of an overview of the process. So what it is, it's building a polypeptide from the mRNA transcript. It's also called protein synthesis, as I mentioned before, where it's in the cytoplasm, and the key players involved are mRNA, the ribosome, and another RNA molecule called tRNA, also known as transfer RNA. So first we'll talk about the ribosome. The ribosome is made of two subunits, a large subunit and a small subunit. These two subunits wrap around the mRNA transcript, so we can see that happening here, and it reads it. And there's three binding sites on the ribosome. There's the A site, the acceptor site, which is where the new tRNA molecules will move in. The P site, which is the peptide site. This is where peptide bonds will form between the amino acid on the P site and the A site. And then the third site is the E site, which is the exit site. So the tRNA that's not needed anymore will leave. And we'll learn more about that through the stages. So tRNA is also known as transfer RNA, and it's called that because it transfers amino acids to the growing polypeptide. So it is a single-stranded piece of the RNA, but it folds onto itself into a cloverleaf shape. So you can kind of see that right here. And you can see there's three different visualizations. This is kind of the two-dimensional structure. This is the more 3D structure, and this would be the space-filled model, showing what it would look like through an electron scan. The tRNA carries amino acids to the ribosome and adds them to the polypeptide. And it has two important parts that we need to note. One of them is the acceptor stem, which is where the amino acid is going to bind. And the other part is the anti-codon loop, which will have the complementary sequence to the codons that are found on mRNA. So a codon is a combination of three nucleotides in the RNA sequence. So the ribosome reads the mRNA transcript 3 nucleotides at a time, and those 3 nucleotides at a time is called a codon. And a codon codes for one amino acid. There can be any combination of the nucleotides, and because there are 4 nucleotides, there can be 4 times 4 times 4 possible codons, so 64 possible codons. But if you remember from Unit 1, there are only 20 amino acids, which means that there's more than one codon for each amino acid. So we end up with multiple codons for the same amino acid, which is actually quite handy because if there is a mutation, it could actually be a silent mutation, and then you would end up not having a harmful effect to the cell. This type of mutation would be known as a silent mutation because it would insert the same amino acid into the protein, and we'll talk more about mutations and more about silent mutations when we do our mutations lesson. So we can read the codon chart by taking a look at the letters. So along the left-hand side, we see that we would look for the first letter. So say it is U, and then we would scan across to the second letter, so UC, and imagine that our thing was UCA, our codon was UCA. So that is found in this section, and the amino acid would be serine. SER is the short form for that. Now, there are some color-coded codons, and those are important to note. Down here, AUG is the codon for methionine, but it's also known as the start codon. So every single protein begins with the start codon, begins with MET. Protein synthesis or translation can't happen until you reach the AUG, the start codon. Then there are 3 stop codons. So when the ribosome is reading across the mRNA and it comes across a UAA, a UAG or a UGA, it will stop translation. So again, if there's a mutation that puts in a stop codon where it shouldn't, then that's going to be really bad for the cell. So translation occurs in 3 stages. First of all, we have initiation. And in initiation, the subunits of the ribosome wrap around the mRNA, so you can see that in this drawing, and it begins reading the mRNA from 5' to 3', one codon at a time. So it's going to scan across, and until it reaches AUG, nothing will happen. So when the start codon aligns with the P site right here, the tRNA with the anticodon, which would be UAC, would bind to the P site. So that would be this tRNA right here. And that tRNA is also carrying the amino acid MET or methionine, which is attached to the acceptor stems right here. At this point, the ribosome will just continue moving along and building the protein. So the second codon for the protein will be found in the A site. So in this case, it's UUU. And a tRNA with the anticodon would bind here. So the anticodon would be AAA. And that codes for the amino acid lysine. So it would bind into the A site. So we can see that here. And then a peptide bond would form between methionine and lysine. And this is going to continue into the next slide. So then the mRNA is going to shift over one codon. So now you can see that the AUG and the first tRNA is in the E site, and then the UUU is in the P site. And now we're going to bring in a third tRNA to the A site. So when that happens, the tRNA that is now in the E site leaves. The third tRNA comes into the A site, and now a peptide bond would form between lysine and proline. So you can see that would happen there. This continues over and over again, so then everything shifts over. The second amino acid or tRNA will leave, and then the next one will come in, and it just happens repeatedly over and over until we reach a stop codon. So the last stage of protein synthesis is termination, and at this point, like I said before, the process will continue until a stop codon is reached. And in this example, our stop codon is UAA. At that point, a releasing factor will bind to the A site, which causes the tRNA to release the amino acid, and then the ribosome subunits and the mRNA and the polypeptide all fall away. So here's our polypeptide, which looks like a chain of beads. So that's our primary structure. That will go off and be folded into the secondary, tertiary, and possibly quaternary structure. And then all the tRNAs fall off, the ribosome falls apart, and then the mRNA transcript leaves, and translation has halted.